Hi, science friends. This is the very last lesson in the genetics unit. We are slated to have a test on Monday. Depending on whether or not we are back to school tomorrow, Friday, will determine whether or not we have our test on Monday. So if we are back to school and can review on Friday, then we will have the test on Monday. If we are not back to school on Friday, and do not have a chance to meet together one more time before our test on Monday, before the test, then we will push the test to Tuesday. So I do want us to have one in-person day before we take that test, just so you'll have the opportunity to ask any questions, and we can go over a few more things and review together. So that's the plan going forward. Today we're going to talk about alteration of chromosome number, though. So sometimes you can look at a karyotype. Remember, a karyotype is the pictures of the chromosomes, and you can see an extra chromosome or a missing chromosome. Now, this can happen on an autosome. Remember, an autosome is one of the chromosomes, number 1 through 22, or it can happen on a sex chromosome, the X chromosome or the Y chromosome. But what happens that results in an alteration of chromosome number is something called non-disjunction. And non-disjunction happens during meiosis when the chromosomes are being pulled apart. And so in non-disjunction, the chromosomes do not get pulled apart properly. So the problem is actually happening in what phase? Think about that for just a second. What phase are they pulled apart? It'll be anaphase, okay? And so if they don't get separated correctly, then one gamete will receive 2 copies of the same type of chromosome, and the other gamete doesn't receive a copy at all. And so then if that gamete goes on to be the sperm or the egg that develops into a child, then they will either not get a copy of one of those chromosomes, or they'll get an extra copy of one of those chromosomes. Now, I believe that we talked about this a little bit in class, but there are some disorders, such as Down syndrome, for example. Down syndrome is trisomy 21. They have a 3rd copy of the 21st chromosome. Trisomy 18 is another one that you hear about from time to time. Individuals are more likely to have an extra chromosome, as in trisomy 21 and trisomy 18, than they are to have a missing chromosome. And the reason for that is these chromosomes code for genes that are really, really important. And so if you're missing a chromosome, that means you're going to be missing out on lots of genes, however many genes are on that chromosome. And so those proteins, your body's not going to be able to produce. And many of them, most of them are there for a reason. They're vital for survival. And so that's why you don't see a whole lot of individuals that are missing whole chromosomes. You don't hear about those disorders because they don't really exist. Those would spontaneously abort. You could have an egg that way, and you could have a sperm that way, and they could combine in fertilization and start to develop a zygote or an embryo. But as that zygote or embryo is forming into a baby, it's going to be missing a lot of its normal life processes, and so it would go through a spontaneous abortion, and it may be so early in pregnancy that the mom may never even known she was pregnant. So that's why you don't see a whole lot of those conditions where you're missing whole chromosomes. Okay, but this condition is referred to as aneuploidy. So aneuploidy is when you have one or more chromosomes in extra copies or deficient in number. Okay, so if you have an extra one, that's aneuploidy. If you have one less, that's aneuploidy. Okay, trisomy 21, trisomy 18, those are all aneuploidy. The only conditions that I'm familiar with where you actually have a whole less chromosome is like Turner syndrome. Turner syndrome, they usually write it as XO. So they're females because they have that X chromosome and they don't have a Y chromosome, but they're missing that chromosome. There's only one sex chromosome there. And so that would be a condition where you have one too few chromosomes, okay? Sometimes you will see them written as 2n plus 1, or you could see them written as 2n minus 1. So remember, n represents the diploid. 2n represents the diploid number. So for humans, that's 46. And so an individual that is aneuploid is going to be 2n plus 1, so 47, or 2n minus 1 would be 45. So sometimes you'll see it written like that. Now, aneuploidy is very different from polyploidy. So look at that prefix. Poly means many, okay? They are going to have many copies of a chromosome, more than 2 complete chromosome sets. So normally we have 2n, right? That's our diploid number. That's what we want our cells to have, except for sperm and egg. They would have the haploid number. But in polyploidy, they could have 3n, or 4n, or 5n, or 6n, and so on and so forth. Now, this is not typical in humans. You don't see this in humans. But what you do see polyploidy in is plants. Polyploidy is very, very common in plants. We didn't extract DNA in this class, but oftentimes in other classes, sometimes I will extract DNA with students. And so you can extract DNA from your mouth, and you get DNA from your cheek cells by rubbing a toothpick on your cheek, but it's not a lot. You just don't see a lot there. But if you extract DNA from a strawberry, you get a lot of DNA. And the reason is because strawberries are naturally, I believe they're hexaploid, okay? Many plants are naturally polyploid. They have lots of copies of their chromosomes. So instead of having 2 copies of the second chromosome, they may have 6 copies of the second chromosome if they're hexaploid, okay? And that's their normal condition. So this diagram is just showing non-disjunction. Non-disjunction can happen in meiosis 1, where these homologous chromosomes don't get pulled apart. And so as a result, this one has extra chromosomes and this one is missing chromosomes. Or it can happen over here in this example in meiosis 2, where the sister chromatids right here don't get pulled apart. So remember, sisters here, and then up here we have the homologous pairs in meiosis 1. And so those would have, some would have extra chromosomes in +1. Some would have not enough chromosomes in -1. Now in this example here, those would actually have the normal number of chromosomes, because this first cell went through meiosis 1 fine and it went through meiosis 2 fine. Just this one here did not go through meiosis 2 correctly. But those are going to be aneuploid here. All of the ones that I've underlined here, those are going to be aneuploidy. These cells show aneuploidy. They have an extra chromosome or they have 1 less chromosome. So that non-disjunction can happen in meiosis 1 or meiosis 2. Always, however, during anaphase when they're being pulled apart. Okay? Now, aside from changing the complete number of chromosomes, you can also change the structure of a chromosome. So you can have a deletion, a duplication, an inversion, or a translocation. And so in a deletion, if this is your original chromosome here, these are showing different genes, A through F. You might have one of your genes, notice that B got deleted, okay? And so a section of that chromosome is deleted. Now, in this diagram, it's very simple. It looks like just the one B gene got deleted. But you could also have a bigger segment of a chromosome get deleted and delete several genes, okay? Now, if you had early prenatal testing and you had a karyotype done, because of the Human Genome Project, because scientists have fully sequenced all the genes in the human genome, if they saw this on your karyotype, they saw that this, let's say, 17th chromosome had a deletion right here, they looked at those chromosomes and they say, hey, this one looks a little smaller than the other, they would actually be able to tell you what genes are going to be missing before the child was ever born, because they have already sequenced all of that DNA and they know exactly what that section is and what is coded for by the part that is missing, okay? So that's kind of some neat technology that we have thanks to the Human Genome Project and the fact that we know all of the DNA and what it codes for humans, okay? So opposite of a deletion, you can have a duplication. So here, B got copied twice, so you would have extra copies of those genes. You can have an inversion where parts of the chromosomes are kind of switched places. So that is an inversion. And then you can also have a translocation where a chromosome that would sit kind of side by side here with it, those two kind of switch spots. And so that would be a translocation, moving one segment of a chromosome to another one. So those are all things that can change chromosome structure as well. Okay? Let's talk about a couple more things here real quickly before we wrap up. So genomic imprinting. There is one thing called genomic imprinting, and I usually love to show this video about genomics, and we'll talk about it more in a unit a little further down the road. But genomic imprinting is when you have a phenotype difference depending on whether you inherit something from the male or the female, okay? If you inherited a certain gene from the mom, it would show a different phenotype than if you inherited it from the dad. And that's what genomic imprinting is, okay? And then lastly, we'll get into this more later on, extranuclear genes. So extranuclear, let's think about what that word means. Let's break it down. So nuclear, we're talking about nucleus. Extranuclear is going to imply outside of the nucleus. So think about this for a second. Where do you have genes? Where do you have DNA outside of the nucleus? There's two places. Think about it. You should say the mitochondria and the chloroplast. The mitochondria and the chloroplast both have their own DNA, okay? Found in the stroma of the chloroplast, found in the matrix of the mitochondria. And so those are also segments of DNA that code for genes that can affect phenotype, can affect genetic traits. Okay, mitochondrial DNA is always inherited from your mother. Okay, mitochondrial DNA is always inherited from your mother. So that's something you need to know there. All right. That is the end of the lesson here. You will have one other short assignment today on Google Classroom, looking at a few karyotypes and identifying a couple of genetic abnormalities based on the karyotype. So that will be submitted on Google Classroom, so make sure to take a look at that. The link is up on the class calendar. So if y'all have any questions, let me know. If not, then have a great day.